Understanding the difference between cleaning validation and biocompatibility testing in medical devices. In the development and regulatory assessment of medical devices, both cleaning validation and biocompatibility testing are critical to ensuring patient safety. Although these two share some features and are closely linked, these processes serve different purposes, follow different methodologies, and are governed by several distinct international standards, such as ISO 19227 and ISO 10993, respectively. Understanding the distinction between them is essential for manufacturers, regulators, and assessors involved in medical device evaluation. Purpose and scope. Cleaning validation, guided by ISO 19227, is primarily concerned with verifying that medical devices, particularly orthopedic implants, are free from manufacturing residues, contaminants, and other unintended substances before sterilization. It ensures that the cleaning processes used during manufacturing are effective and reproducible. In contrast, biocompatibility testing, as defined in the ISO 10993 series, assesses the biological safety of a medical device when in contact with the human body. This includes evaluating the potential for toxicity, irritation, sensitization, carcinogenicity, and systemic effects, among others. It considers not just residues, but all components and interactions of the device that may affect patient health. Methodological differences. ISO 19227, Cleaning Validation. Focus: verification of cleaning procedures for orthopedic implants. Methodology: involves establishing acceptable limits for residues, example, detergents, oils, particulates, following a risk-based approach. Specifies methods for sampling, extraction, and quantitative chemical analysis, example, total organic content, TOC, or total hydrocarbons, THC, or gravimetric analysis. Emphasizes repeatability, worst-case conditions, and risk-based limits. Typically done before sterilization, focusing on the manufacturing process. End goal: demonstrate that devices can be consistently cleaned to acceptable safety levels prior to sterilization. ISO 10993, Biocompatibility Evaluation. Focus: comprehensive assessment of the biological effects of a device or its materials. Methodology: risk-based approach as described in ISO 10993-1. Includes chemical characterization, ISO 10993-18, toxicological risk assessment, ISO 10993-17, and biological testing, e.g. cytotoxicity, irritation, sensitization, systemic toxicity. Requires evaluation of extractables and leachables, often using extraction protocols, sometimes under exaggerated conditions. Testing is usually conducted post-sterilization and considers the full clinical use scenario. End goal: ensure that the device under actual use conditions is biologically safe for patients. Conclusion: while cleaning validation and biocompatibility testing may both deal with substances that can pose a risk to patients, they serve distinct and non-substitutable roles. Cleaning validation, ISO 19227, ensures that devices are properly cleaned before being sterilized and used, while biocompatibility testing, ISO 10993, ensures that the final sterilized device is safe in the intended clinical context. While closely linked, one cannot replace the other, and regulatory compliance requires a clear understanding of both. Professionals evaluating medical devices must be trained to differentiate these methodologies to ensure comprehensive patient safety.